Hello, and welcome to this video on nature-inspired marine anti-fouling surfaces. In this video, an overview of marine biofouling will be given, as well as the problems that it causes. Nature-inspired anti-fouling surfaces will also be discussed, including the different types that currently exist and how effective these are. During this video, several different topics will be discussed. Firstly, the concept of biofouling will be introduced. Second, bacterial adhesion and biofilm formation. Third, some common marine fouling organisms. Fourth, marine anti-fouling strategies. Fifth, natural anti-fouling surfaces. Sixth, the production methods used to produce these anti-fouling surfaces. And seventh, the testing of nature-inspired marine anti-fouling surfaces. And so this first section of the video gives an introduction to biofouling. Biofouling is the process by which undesirable or unwanted biological material builds up on surfaces. The process starts when organisms and biological material present in the fluid settle on the surface. When organisms settle on the surface, they then begin to reproduce. High levels of organism attachment then begin to create problems for the surface. Biofouling leads to major economic and environmental impacts in a variety of industries. One of these is the marine industry, including both shipping and aquaculture. For shipping, the biofouling of cargo ship hulls significantly increases drag, which leads to greater fuel consumption and larger volumes of CO2 emissions released. Biofouling also causes problems in the medical industry, as the fouling of medical devices and hospital surfaces by microorganisms leads to an increased risk of infections. Biofouling of dairy pipelines and food packaging causes product contamination and reduces shelf life. Oil rigs and pipelines suffer from increased rates of corrosion due to biofouling. This increases costs as equipment must be regularly cleaned or replaced. In water treatment plants, the biofouling of treatment membranes and sewage pipes leads to decreased water flow rates through these pieces of equipment. The biofouling process varies depending on the setting in which it is occurring. This is due to the different conditions and fouling organisms present in each environment. This video will now focus on marine biofouling. The marine biofouling process occurs in 5 key stages. The first of these is the formation of the conditioning film, which forms on a surface within a few minutes of submersion. After a few hours, microscopic organisms begin to attach. The population increase of microorganisms results in the formation of a biofilm. Juvenile macrofouling organisms then begin to adhere to the surface. And finally, after a few months, a full biofouling community is formed. The next section of the video will discuss the topics of bacterial adhesion and biofilm formation. One of the first stages of marine biofouling is bacterial adhesion and biofilm formation. Biofilms attract larger fouling organisms which prefer to settle on organic material rather than pristine surfaces. Biofilms also increase rates of corrosion through a process known as microbial influenced corrosion. This can lead to damage to the material surface, as shown here. They also reduce hydrodynamic efficiencies of ships. Depending on the thickness of the biofilm, the power requirement to move a mid-sized naval vessel can be increased by up to 10%. The formation of a biofilm can be broken down into 5 key stages, shown in the flowchart here. Here, the graph on the left shows how the population of bacteria on a surface changes with time. The area highlighted in red refers to the stage currently being discussed. The first stage of biofilm formation is the establishment of a thin conditioning film. These films consist of sticky organic material such as proteins and polysaccharides that are present in natural water. These large organic molecules adsorb onto a surface almost immediately after immersion into the water. Areas of charge density caused by differing concentrations of charged amino acid groups help microorganisms reach and attach to the surface. After the conditioning film has formed, the reversible adhesion stage of biofilm growth begins. In this stage, microorganisms such as bacteria and diatoms are transported from the bulk fluid to very near the surface. Some use appendages such as flagella to propel them towards the solid, whereas others rely purely on turbulence. At this point in biofilm formation, microbes can still easily detach from a surface if they sense that it is unsuitable. Adhesion transitions from reversible to irreversible when adhesins are released. Adhesins strengthen the bonds between the organism and the surface, whereas the reversible attachment is fundamentally controlled by physical forces such as van der Waals and electrostatic interactions. Irreversible attachment is controlled by much stronger chemical interactions. The strength of the attachment is governed by the material's chemical and physical properties. The permanent attachment of microorganisms to the surface allows for reproduction and population growth within the biofilm. In this stage, large quantities of extracellular polymeric substances, or EPS, are secreted. These act as a house for microbes, protecting them from harsh environments. The EPS matrix also binds together the biofilm and allows nutrients to be transported throughout the population. As biofilms mature, dispersal becomes an option. This can occur passively as individual cells become accidentally detached from the biofilm structure, or it can occur actively as cells seek to remove themselves from the biofilm. Active dispersal occurs if microorganisms perceive through environmental changes whether it is beneficial to escape the biofilm structure. When this happens, cells release enzymes to break down the EPS matrix, allowing them to free themselves and search for new surfaces. In the third section of the video, we will take a closer look at some of the organisms that are responsible for biofouling. In this section of the video, some common marine fouling organisms will be discussed. These organisms can be divided into 2 categories. The first are microfouling organisms, which measure only micrometers in length and include bacteria, algal spores, and diatoms. These organisms are responsible for the biofilm growth discussed in the previous section. Then we have macrofouling organisms, which are much larger and more visible. Due to their larger size, the detrimental effects of macrofoulers are often more serious. Some common macrofouling organisms include algae. Algae can occur both as light algal slime or much larger macroalgae, such as seaweeds. Algae require light, and so their habitat is limited to depths to which light can penetrate. Bryozoans. Bryozoans are filter-feeding invertebrates that form colonies on submerged surfaces. They are among the first colonizers of new or pristine surfaces. Mollusks. Mollusks, such as mussels, can attach to ship hulls and are then transported around the world where they outcompete local populations. Tubeworms. Tubeworms' chalky exteriors form crusts on boat hulls, which roughens the surface and significantly increases drag. Barnacles. Barnacles are regarded as the most problematic biofoulers due to their prevalence and ability to strongly adhere to submerged surfaces. Here, a closer examination of green algae species, such as Ulva linsa, shown here, is undertaken. These species are commonly found in the upper intertidal zone of coastlines. They are often used as model marine fouling species as they can be readily found on man-made structures and ship hulls. They settle on new surfaces through the release of pear-shaped zoospores, which travel through the water column until they settle on a surface. They then produce an adhesive pad to ensure firm attachment. Barnacles, such as those shown in the image on the right, are another example of marine fouling species that is commonly used as a model organism. Juvenile barnacles are called cyprids and measure only millimeters in length, as shown here. Cyprids represent the stage in the barnacle life cycle in which surface attachment occurs. A cyprid will first explore a surface using 2 feet to assess whether it is suitable for attachment. If suitable, the cyprid will then release a dopa-rich cement, which permanently binds the barnacle to the surface, where it can then mature. The level of adhesion between organisms, such as those that have just been discussed, and microtextured surfaces can be described by attachment point theory. This theory is based on the fact that when the area of contact between organisms and a surface is larger, the adhesion is stronger. Organisms will prefer to settle on sites where they are strongly attached to the surface, and so the number of potential settling sites will depend on both the size of the features of the surface and the size of the settling organisms. Moving on to marine anti-fouling methods that are currently in use. Throughout the mid-20th century, the most common way to prevent biofouling was to apply tin-based paints to surfaces. These paints are now banned as they damage both coastal and marine ecosystems, and so new anti-fouling strategies are required to prevent biofouling. These strategies can be divided into 2 categories. Biocidal surfaces are toxic to organisms that settle on them, and so prevent attachment. Fouling release surfaces prevent settlement as organisms can only attach very weakly and so are washed off by water currents or other dynamic forces. A good biocidal anti-fouling coating, with respect to the environment, is effective in preventing fouling of the painted surface without persisting at concentrations greater than those that can cause detrimental environmental effects. Common ingredients of biocidal anti-fouling coatings include copper, shown here, and relatively environmentally friendly biocides such as C9, the chemical structure of which is shown on the right here. Fouling release coatings generally have low surface energies and high elasticity, meaning that organisms can only weakly attach. These coatings are commonly silicon-based, with PDMS, shown here, often used. Any organisms that attach to the coating are easily removed through hydrodynamic shear forces at the surface, which, for example, can be created by ocean and tidal currents, or by the movement of a ship. As they do not contain biocides, these coatings are generally considered to be more environmentally friendly than the biocidal coatings. The upcoming section of the video examines antifouling surfaces that occur in the natural world. In this section of the video, we will take a look at some nature-inspired marine antifouling surfaces. The surface of plants, such as lotus leaves, and animals, such as shark skin, have evolved remarkable antifouling strategies over millions of years. By studying these surfaces, new man-made antifouling strategies can be developed. As mentioned, the lotus leaf has evolved a remarkable antifouling strategy. The surface of lotus leaves are both rough due to a covering of microscopic pillars and have low surface energy due to the presence of wax crystals. The combination of a high surface roughness and low surface energy leads to superhydrophobicity and air being trapped between the water and solid surface. This leads to water droplets rolling off the surface easily. As they roll, the droplets pick up dirt and organisms that may be fouling the surface. Another family of plant species that display superhydrophobicity are the Salvinia water ferns, shown in the image here. The layer of air that is created when water rests on superhydrophobic surfaces can act as a physical barrier, preventing any fouling material or organisms from reaching the surface. The Salvinia water ferns have been known to trap layers of air for months at a time when the leaves are submerged in water, whereas the lotus leaves do not produce stable air layers when submerged. The long hairs of the Salvinia plants, pictured here, are thought to aid in trapped air layer retention. The pitcher plant, shown here, has also inspired the production of antifouling surfaces. The inner walls of this plant trap a layer of fluid within the surface microstructure, creating an intermediary liquid layer that reduces adhesion between objects and the surface. For the plant, this means that insects that wander into the inner section slip down the walls and cannot escape. They are then digested. Liquid-infused surfaces, such as the inner wall of the pitcher plant, are termed slippery liquid-infused porous surfaces, or slips. Slippery surfaces, such as this, have been shown to prevent up to 99.6% of bacterial attachment. Gas trapping and slippery liquid-infused porous surfaces can be used to prevent the attachment of marine fouling organisms, as the gas or liquid layer prevents the organisms from reaching the solid surface. In order to preserve valuable energy, sharks have evolved to be streamlined. The fouling of their outer surface by other organisms would increase drag, which would therefore mean the shark would need to use more energy to swim. Therefore, shark skin consists of small protruding dermal denticles that provide them with both drag reduction and anti-fouling properties. The anti-fouling characteristics of the shark skin are due to the roughness created by the tiny riblets. Patterns inspired by shark skin have been manufactured to be applied as a coating to medical surfaces such as catheters and wound dressings to prevent microbial attachment and therefore infection. Some mollusk shells have adopted a similar anti-fouling strategy to shark skin that is also based on the roughness of the surface. The shells of the mussel species such as Mytilus edulis and Mytilus galloprovincialis have a ridging structure that covers the outer surface of the shell. These ridges are around 120 nanometers high and have spacings of around 1.8 micrometers. This makes it difficult for microorganisms to attach to the surface as the area of surface contact is reduced. In order to reduce the risk of disease and preserve energy, fish have developed anti-fouling strategies to deter organisms from settling on their skin. In fact, the structure of fish scales is arranged such that it retains a hydrophilic lubricating gel that is excreted from glands in the skin. This gel reduces the coefficient of friction of the surface and makes it difficult for organisms to attach, and also reduces drag. The next portion of the video reviews selected production methods that have been used to manufacture nature-inspired marine anti-fouling surfaces. Some common production techniques that can be used to produce nature-inspired anti-fouling surfaces will now be discussed. The first of these is 3D printing. 3D printers, such as this one here, are an increasingly common production tool to produce microtextured and nature-inspired surfaces. They are flexible, cost-effective at small scale, and the designs can be tweaked and updated easily using computer-aided design software. This makes them very useful for research, where they can be used to produce a range of surface topographies with different materials, or even with a combination of materials. They can therefore be used to engineer substrates to have specific physical and chemical properties. However, many commercial 3D printing technologies lack the printing resolution required to produce micro or nano-textured surfaces. Laser beams are very narrow, have high energy, and can be focused onto a small spot, allowing them to produce a wide range of surface textures. With the development of pulsed femtosecond laser processing, micro and nano-textured surfaces can be manufactured at large scale. The short duration of laser pulses also reduces thermal damage to the surface, and so laser processing can be used on a wide range of materials, including metals, ceramics, and polymers. However, laser processing machinery is expensive, and due to the nature of the process, overhanging structures such as salvia leaf replicas cannot be produced. Another way to produce biomimetic anti-fouling surfaces is by using lithography-based techniques. These processes are often used to produce micro or nano-textured surfaces due to their high accuracy and ease of scalability. The process starts by coating a surface with a photoresist, onto which a photomask is then placed to cover specific areas. The surface is then exposed to UV light or lasers, which interact with the uncovered areas of photoresist. Patterned areas of photoresist can then be used to create a mold, from which repeated patterns can be quickly manufactured. A downside of lithography-based techniques is that if the design needs to be updated, then a new master mold has to be produced. This is both time-consuming and expensive. This final section of the video will discuss the methods by which nature-inspired anti-fouling surfaces are tested. To test anti-fouling surfaces, they must be placed in environments in which fouling organisms will try to settle on them. Field testing involves transporting test samples out into the ocean or to a freshwater body and leaving them there for extended periods of time. This method is useful as it replicates the fouling stresses that the surfaces are under when they will be in use. However, field tests are expensive, both in terms of time and money, especially when specialist equipment such as submersible cameras and scuba gear is required. Also, they take place in an uncontrolled environment, and so comparing samples that have been tested in different locations is difficult. Anti-fouling surfaces are also tested in laboratory-based experiments. These experiments are conducted in a controlled environment and are completed quicker than the field tests. They are therefore useful for quickly comparing the anti-fouling capabilities of two or more surfaces. They can also be conducted cheaply and from anywhere in the world, even far from the ocean or natural water, as long as the right equipment is available. However, as laboratory-based anti-fouling studies only test surfaces using a small number of species, the effectiveness of these studies doesn't necessarily translate into the anti-fouling properties in real-world conditions. This video has highlighted the problems that biofouling creates for a number of different industries. It has discussed the processes by which biofouling occurs, including biofilm formation and also some common marine micro and macro fouling organisms. Natural surfaces such as salvinia leaves and shark skin have inspired the production of anti-fouling materials. A variety of techniques, including 3D printing, laser processing, and lithography-based techniques, can be used to manufacture these biomimetic surfaces. The effectiveness of these surfaces can be tested in both field and laboratory-based experiments, although it is still a major challenge to find anti-fouling surfaces that are both environmentally friendly and effective against all species.